So, as I said that this is the topic very rarely people discuss, and we talk about the viruses, those viruses which are harmless, those viruses for which we do not have much information, but they are very much here. So you see that when I ask you guys, I'd like to make it more interactive, so in between you can ask things, rather than just waiting when the entire lecture is over, then we interact. You can stop me in between, we can have a good discussion. So when I ask you that, tell me the number of viral diseases that you are aware of. Not the naming of the virus as such, but how many viral infection you think there could be. Any guess, any guess. Aap bataएं?
Measles.
No, number, like 10 infection, viral infection, 20 viral infection, approximately how many viral infection you can think the person may have? कुछ भी कोई, this is not an evaluation criteria, कुछ मतलब I just want to know what you have in your mind. How about 100? Anyone else? 2000, let's say 2000. और कोई yes? कुछ भी बोल दीजिए, आपका कम से कम वोट तो काउंट हो जाएगा. तो इन्होंने बोला कितना? 100, and other person, your colleague said 2000, right? So let's say 5000, ठीक है? So that it includes many viruses around that causes infection. But if there are, let's say, 5000 viruses that are able to cause infection, then we should know how many viruses are around. Ever thought about the number of viruses that are around? So how many viruses exists in the nature? अगर 5000 infection cause कर रहे हैं, तो do you think that there would be like, more than 5000 viruses? What is your guess? You see that approximately 10 to the power 31. So this statement is interesting. There are more viruses than stars in the universe. 10 to the power 31. And if you say that 5000 are causing infection, what percentage of viruses are causing infection? Negligible, right? I haven't calculated the percentage, but it will be very, my 0.00 something, It means that all the viruses that are around, they're not doing anything to us. And we do not know whether they are not doing anything or they are helping us to survive. You see that? That's the interesting observation, and this can be a really interesting topic to do research work to find out what other viruses are doing. Anyway, so what is the special nature, or what is so special about the viruses? First thing we should know that they came into the existence around 30 million years ago. 30 million years ago. It's long back. We all are exposed to various kind of viruses, as I said, that 10 to 31, and we breathe viruses, we eat viruses, every time, whatever activity we are doing, we are exposed to the viruses, right? We agree with that, right? Only few are pathogenic, because if there are so many viruses, and if saying that only 5000 are causing infection, so I would say that only few viruses are causing the infection, right? Interestingly, there is no treatment of viral infection available so far. Few vaccines have been generated, but important thing is that whenever you have viral infection, you go to the doctor, they say take rest, hafte bhar mein you just take paracetamol. Whenever you have high fever, you develop immunity and then you get rid of that infection, correct? Very rarely you end up getting the antiviral therapies, in special cases, but for those infections for which you get antiretroviral therapy like HIV, which I will be talking about, you don't end up getting cure. They suppress the virus replication, virus remains inside. So this is something I can say that no treatment available. For no cure is available, but treatment is there. So it should be read like no cure is available. You cure the viral infection by yourself. Body clears that by themselves. Only vaccines are effective. So what is the role of other viruses in our life? This is important thing we should know. Before we talk about it, first thing, we discuss this entire lecture in two different parts. One part is that this junk DNA, which we have, made up of transposable element. You must have studied about it, right? transposable element that transpose from one location to another locations, right? Some transposable element, they rely on RNA. What happens? These transposable element through the process of transcription, because these are RE, retro element, through the process of transcription, they make RNA copy, and then they could convert back into the DNA using reverse transcriptase, and then this DNA transposed to some other location. Like jumping gene concept, the Barbara McClintock, right? The same principle is there. Now retro element, initially it had one copy, and now it has become two copies. When you truly investigate different kind of retro element inside our body, then surprisingly you get to know approximately half of our genome. Here I'm just writing 45% of our genome is composed of retro element, transposable element that transpose from one location to another location. So there are two ways. Either they transpose through the DNA itself, DNA doesn't get converted into RNA, or they get converted into RNA and just get inserted in some other location. So now see that if they are actually transposing as a DNA itself as an intermediate, it is called transposons. That's what actually in May's Barbara McClintock jumping gene concept was given, right? Now there are certain transposable elements that transpose from one location to another location using RNA as an intermediate, as I described in the previous slide. When this happens, then we need to know what these are. They are called retro element. Here what we said, transposons. Here what I'm saying, retro element. If you look at the retro element sequence, and then you find that there are certain retro elements which do not have long terminal repeat like sequence. You know about long terminal repeats? Anyone? Long terminal repeats are a unique sequence you find on the 5' and 3' sequence of HIV virome. HIV का nucleic acid अगर आप लें HIV RNA, when it gets converted into DNA, then you have a special 5' or 3' sequence. They are called long terminal repeat. So any sequence that resembles with a long terminal repeat, we can say that retro elements having LTR containing sequence and retro element containing non-LTR sequence, because there is no LTR, right? कुछ बोलिएगा तो मुझे मालूम चलेगा I should proceed further or I should make it clear to you at this point, right? So when there are non-LTR containing sequence or retro element, we call sign sequence, ALU sequence. Molecular biology you must have studied. Sign sequence, ALU sequence, right? Pseudogenes, only what percentage? 33.9% or something like that. Majority of retro element fall under this category. There are certain other categories for which we have little information so far, which I would like to introduce you about it. It's called LTR containing retro element. LTR, same sequence having resemblance with that of HIV genome, right? They are called LTR containing sequence. These slides are pretty old. It is saying 8.3%. But currently, there are approximately 14% of our genome is made up of retro element which contains LTR containing sequence, and these are known as human endogenous retroviruses. In short, we call it as HERV, H-E-R-V. Ever heard of this name? Yes or no? Yes. Yeah, HERV. So HERV, LTR containing sequences. Now what are these HERVs? As I said that approximately 14%, 8 to 10% is written, but later on it was confirmed approximately 14% of our genome, we all are having sequences resembling to the retroviruses. These are called human endogenous retroviruses, how it got into our genome. So they are remnants of ancient retroviral infection. Long time back, let's say million years back, we got infected with the retrovirus. That retroviruses, when infected a person, got somehow mutated, got integrated in our germline, we cleared the infection. And from one person to another person, since it got incorporated in our genome, it got transmitted from one person to other person. That's what we call vertical transmission. Those who could not fight with this retroviral infection, they were eliminated. Maybe they did not survive. But those who survived had this human endogenous retrovirus in their genome. All we are sitting here, we have 14% of genome component made up of retroviruses. So viruses are there with us. We are with the viruses. Nothing is happening. So majority of HERV in human genome exists in truncated form. It has to be truncated. It is not replicating and shedding the viral particle because it has undergone several mutations. They are just existing as a latent viral or something like a truncated viruses which are not able to transcribe and translate into the new viral particle. That's it. But they are very much here. We did not bother about it because they're not doing anything. As I said, that junk DNA concept, but later on, new story evolved, which I'm going to tell you about it. There are more than 40 different families of human endogenous retroviruses. So if you compare these human endogenous retroviruses with the rest of the retroviruses, so you see that they fall into 3 different classes. Class 1, Class 2, Class 3. And you can see that they are either HERV-K, HML2, we also call it, HERV-H, HERV-W, HERV-L. These are the different classes of human endogenous retroviruses. I'm not getting into the strict classification of these human endogenous retroviruses, but I'd like to tell you what about their biological role as such. So when I try to compare between retroviruses or endogenous human retroviruses, so we find that basic retroviral genome, if you have studied, it includes 3 structural genes, GAG, POL, and envelope. Upstream of GAG, you have U3, RU5 region. All together is termed as LTR. Likewise, on the 3' side, you have U3, RU5 as well. This is also called LTR, long terminal repeat. This is a gamma retrovirus genome, right? When we compare it with human endogenous retrovirus, what happens? We have either the complete genome or solo LTR. What does it mean, solo LTR? 2 LTR combined with each other and they make solo LTR. But this genome has several mutations, either point mutations, and you see because of these mutations, what will happen sometime, suppose you have mutations in POL and envelope, only GAG is there, which is intact. So what happens? GAG has ability to undergo the process of transcription and translation because GAG here is not mutated. I'm just giving you an example. Likewise, assert because it has capability to transpose a different location in our genome. So at certain position, you will have intact envelope. So what will happen here? Envelope has capability to transcribe and translate. We do not know which is going to get transcribed, which is going to get translated. It's just by this figure showing the possibility. That's it. But as we further evolved with our research on human endogenous retroviruses, we came to know that HERVs have association with certain diseases. What kind of association it is? So if you look at first, different kind of autoimmune disorder, let's say the person is suffering with rheumatoid arthritis, lupus, multiple sclerosis, Aicardi-Goutieres syndrome, people started noticing that there is an upregulation of RNA transcript of HERV, either envelope or GAG or POL, not the entire virus, only small part of it. So people were wondering why there is an upregulation of such kind of human endogenous retroviral genome or certain genes. So 2 things. When transcription is happening, you get RNA. If RNA get translated, you get protein. And if by chance this protein is expressed on the surface of the cells, so there is a possibility that your immune system, which has never seen this protein before, can get activated and find it or sees it as a foreign protein, and you develop the immune response. So the hypothesis says that if it really happens, then you develop T cell response against that HERV protein. And if there is a T cell response against that protein, there is a possibility that you end up developing antibody response as well. When certain things are happening this direction, it will lead to a condition of autoimmunity, right? This is what actually your own cells is actually expressing.
I have a question.
Yeah.
We know that during the T cell maturation, some part I don't remember that in this moment.
I can tell you what you are answering. You are talking about the tolerance, T cell tolerance in the thymus. You are saying tissue-specific antigen, which is expressed by your medullary thymic epithelial cells, right? Exactly. So these are poor HERV proteins which have never been expressed there, because these are part of junk DNA. They are not supposed to get expressed. Later on, few people found that these are expressed in certain conditions. So that is why it is associated with the autoimmune diseases. So far, people just saw the association. If you have autoimmune diseases, there is a possibility that one of the HERV protein, depending upon what kind of autoimmune disorder you have, you may have expression of HERV protein. So far, we did not know, what is the biological relevance of these HERV protein, what it is doing. It's just simply the expression. Later on, people found that a person is suffering from different kind of cancers. You have increased RNA transcripts of human endogenous retroviruses in the plasma. So many times people also try to, make it as a marker for the malignancy. So if there is a high expression, but it may not be absolute, because as I just previously said, that it can also be expressed during autoimmunity. So you may misdiagnose. So whether you have autoimmunity, you have a cancer. But the fact is that in melanoma, breast cancer, pancreatic neoplasma, testis cancer, teratocarcinomas or seminomas, all these conditions, you have upregulation of human endogenous retrovirals gene. Not the entire virus again, because other part is highly mutated. So either GAG or envelope will be expressed, and when it is expressed, so the possibility is that you will also end up having immune response against it. So far, so good, right? Now the interesting story. Based on these things, we previously had thought what could happen whenever there is an infectious diseases. The infectious diseases which I have been working on is HIV. And HIV is a retrovirus. Human endogenous retrovirus are also retroviruses. They are sleeping inside us. So when HIV enters, what will happen? So you see that we thought that if in cancer or infection or autoimmune disorder, if HERV expression can be upregulated, why not in case of HIV? But before we could actually say that there is an upregulation of HERVs in the setting of HIV infection, we needed to come up with some hypothesis. Hypothesis was based on this. When HIV infection is taking place, it is not taking place in the vacuum. It is happening in the cell. And cell has got what? Cell has got nucleus. And nucleus has got human genome. And human genome has human endogenous retroviruses. So you see that? Exact situation is like that. But now think about this particular cells where HIV is not yet infected. What will happen? HERV is not expressed. HERV can possibly be masked by the presence of certain restriction factor. One of the restriction factor you'll come to know later when you study this retroviruses in future. It's called ApoBac. What ApoBac is doing? ApoBac is restricting the expression of human endogenous retroviruses completely. We are all fine. HERVs are there, everyone's body. But what happens when HIV infects these cells? HIV has got very interesting accessory protein which we call virion infectivity factor, VIF. VIF counteracts the action of ApoBac. So it means that when HIV infects, its virion infectivity factor will simply neutralize ApoBac. When there is no ApoBac, then HERV thinks that no, there is no barrier for me. I can transcribe, I can translate. So then HERV can be expressed, and it is expressed. So thinking this concept in mind, the people back in San Francisco, they did some work. They took samples from HIV-infected persons, plasma sample, they extracted RNA. They also take a sample from healthy individuals, and after extracting RNA, they did PCR study. And the PCR study, what they did, they looked for particular HERV envelope region. And what they found, that in HIV-infected subject, there is an upregulation of human endogenous retroviruses compared to the HIV-negative subject. That came in PLOS Pathogen 2007. That's the picture from that particular paper. So that indicates that HIV infection is causing upregulation of human endogenous retroviruses. Here the story begins. It means that it was more or less confirmed that there is an upregulation of HERV transcript in the setting of HIV infection. In primary HIV infected subject, those who get HIV infection like within 2-3 weeks of time, and they have symptoms of acute HIV infection. So the next question is that if there is upregulation of RNA transcript, so possibly they should make protein as well. When they make protein, then you also develop T cell response, because T cells sees them for the first time, they find it as a foreign protein. Now, there is an assay which we call ELISpot assay. You heard of it? Yeah. ELISpot assay, what it does, suppose a person got infected with any viral infection and the person develops a memory T cells. Now, when this memory T cell again sees the same pathogen, it immediately acts and releases interferon gamma. That interferon gamma you detect. If the person was never got infected with the same virus before, then there would not be any memory T cells, right? So suppose a person got infected with HIV and developed memory cell response. HIV is still persisting in the blood, but memory cells have already, formed. And assume that the person may have also, upregulated HERV expression, and HERV protein was also formed. So in addition to HIV-specific memory T cells, the person would also develop HERV-specific T cells, HERV-specific memory T cells. We do not know. That's a hypothesis. But if I take the blood sample of that person and want to test whether the person has HERV-specific T cell response or not, what I will do, I will purify the PBMC. PBMC includes T cells, memory T cells as well. And then what I'll do, I'll take the autologous cells and infect it either with the whole virus or a small peptide which correspond to the antigen level of either HERV or either HIV. I'll pulse it. That protein or that epitope, that small peptide goes inside antigen presenting cells or target cells, present it together with MHC class 1 molecule, and the PBMC or PBMC sample which I purified from that person, I'll add there, and if memory T cells against HERVs are there and sees any HERV peptide being presented, because I pulse it in autologous cells, and then it interacts and releases interferon gamma. So that particular cell that releases interferon gamma, it is captured by antibodies. We have capture antibody, cells, followed by detection antibody, and we develop a blue spot. We get the blue spot. The number of blue spot we count, we can say number of spot forming unit per 1 million of PBMC that we add. So higher the number of a spot, higher magnitude will be for HERV specific T cells, right? So this way, what had happened, the experiment was conducted. ELISpot assay was performed, as I said. Now, peptide was designed because for HERV, the whole virus cannot be taken, right? We know the HERV sequence. From the GenBank database, we take a small sequence. We put it in this site. It's called NetCTL. NetCTL allows you to form several sets of peptide unique to a particular HLA type. Here I'm not discussing more about the HLA typing and all that. We just assume that suppose I'm targeting one person and one person particular HLA type is, let's say, B51. So this NetCTL allow me to generate peptide which is restricted to B51 allele, so that there is no HLA based biasness, or because of HLA type, I will not miss any response. So we developed a lot of peptide. You see that these peptides says that HERV peptide similar to HIV, HIV peptides are there, and HERV peptides. These HERV peptides are unique to HERV. They do not cross-react with HIV. So whatever response I get using these peptide, they will be unique to HERV-specific T-cell response. All peptides were restricted by HLA class 1 allele. You see this particular graph. Here what it says that there are two categories, HIV negative, empty circle or white circle, and these black circles are HIV positive. So you see that if I just focus on those peptides which are HERV peptide, unique to human endogenous retroviruses, you see that there is a upregulation of these responses. There is a significant higher magnitude of T-cell response against HERV in HIV positive individuals. You see that compared to HIV negative. What does it suggest? It suggests that those people who are HIV infected has also developed T-cell response against human endogenous retroviruses. So you see how story is coming up. Transcription increase, T-cell response. Now if there are T-cell response in HIV patients against HERV, what these T cells are doing. So before we could actually find out what these T cells are doing, we should also do one small study. A small study is nothing but a correlation study. What correlation study suggests? You see that on x-axis we have HIV plasma viral load. How much viral load we have in the plasma. On y-axis, we have HERV-specific T-cell response. It is called interferon gamma spot-forming unit per million of PBMCs. What it suggests? It suggests the indirect correlation. It means that if we have greater T-cell response against HERV, we should expect low plasma viral load, HIV viral load in person. So it means that two persons are there. One person is having very high HIV plasma viral load in the blood. The person may have low T-cell response against human endogenous retrovirus, while the other person who has low HIV plasma viral load, the person may have higher HERV-specific T-cell response. So what message we get? T-cell response against HERV, which people develop during HIV infection, may be beneficial because higher T-cell response, lower HIV load. But we just move forward with the story. One of the important criteria during HIV infection is CD4 cell count, because during HIV infection there is a sharp decline in CD4 count. And when there is a decline in CD4 count, then what will happen? There will be a lot of possibility of having opportunistic infection, because CD4 cells are important as far as our cellular immunity is concerned, right? So, okay, it may come later. So there was also a direct correlation between CD4 count and HERV-specific T-cell response. Somehow, maybe that slide comes later, but conclusion from this part of the study which comes is HIV induces HERV transcription in primary HIV infection. These are primary, there are three different categories of HIV. Primary infection, they get symptoms right after the virus enters in the body, let's say 3 weeks after the infection, that is primary. When we have symptoms goes down and then person lives with HIV for 10 to 12 years. That phase is referred as chronic HIV infection. And third category, which is generally is a perinatal HIV infection. So whatever I'm showing or talking here, three different categories. One is primary HIV infection, second is chronic HIV infection, third is perinatal HIV infection. Now, that's what we say, that HIV induces HERV transcription in primary HIV infection. HERV-specific CD4 T-cell responses are present in primary HIV infection, and HERV T-cell responses are indirectly correlated with the viral load in primary HIV infection. HERV T-cell response exists very early in HIV infection. These are important points, but we do not know what happens if person lives with HIV for tens of years. That is also very crucial phase of HIV infection. So the second part of discussion is, like, once we have discussed about primary HIV infection, what happens in pediatric HIV infection. Like primary HIV infection, primary infection in pediatric HIV infection also, there was a indirect correlation suggesting that both in children as well as adult with the primary HIV infection having the same kind of indirect correlation with HIV plasma viral load and T-cell response against human endogenous retroviruses. Now, important categories. So this is the conclusion of this perinatal thing. So, people think that in early stages of your life, when you are exposed to certain thing, you get tolerized to that. Tolerance develop. But since we also got HERV-specific T-cell responses, so what we know that HERVs are not tolerized in utero. And vertically HIV infected children have HERV T-cell response exactly like primary HIV infection, and a significant indirect correlation between HERV T-cell response and HIV viral load in vertically HIV infection. These important points are also there in case of perinatal HIV infection. Now, chronic HIV infection is a very, very important one, because most of the HIV infected individuals that you see in India is approximately 2 million or 2.5 million people living with HIV. So, since they are living with HIV, they are referred to as chronically HIV infected patients. What is happening in them? So, to investigate differences in HERV T-cell responses among adults with chronic HIV-1 infection, here the study was conducted. In this study, you see that it's an old study where the different categories of HIV infected people were taken. You see there are controller, there are virological non-controller, immunological progressor, heart suppressed. What is heart suppressed individual? Those HIV infected people who start antiretroviral therapy, they suppress the viremia completely. Or you can say that they have undetectable viral load, because anything below 50 is referred to as undetectable because of the sensitivity of the machine. If they are on effective antiretroviral therapy, it is also called suppressive therapy because it is suppressing. Other category of people is that those people who control the virus. There are certain category of people, even without medication, they control the virus. They are called controller. In general, they are referred as elite controller. But those who are taking, they are other untreated categories are those HIV infected. We have one we call virological non-controller. It means that the virus is high, but CD4 count is still preserved. Immunological progressor means even their CD4 count is low. So different category of people are there. HIV negative subjects are also there. When we looked at T-cell response against HERV in all these categories, you see that controller who control the virus by themselves have the highest magnitude of HERV-specific T-cell response. They are controlling. Other category, which we can say hard suppressed, those who are taking antiretroviral therapy and they're suppressing the viruses, they are also having higher magnitude of HERV-specific T-cell response compared to other category. You see non-controller and progressors. Progressor who progress to HIV, they have high load. You see that they have low HERV-specific T-cell response, almost compared to the negative one. So the presence of high magnitude of T-cell response against HERV in controller is again suggesting that it helps in controlling the viremia in some ways. So HIV controllers have the strongest HERV T cell response. This is the take-home message, right? If you just look at the controller and try to find out whether they are still showing indirect association between HIV-1 viral load and HERV-specific T cell response, indeed, they are having inverse correlation. Higher the magnitude of T cell response against HERV, lower will be the HIV viremia in the plasma, right? And now, first thing which I was mentioning about the CD4 count. Now, CD4 is very, very important. If it goes down, then you develop opportunistic infection. Actually, in AIDS as well, what happens, CD4 count really goes down, and then you have a problem, viral load is high, and you get opportunistic infection. But here you can see that on x-axis we have CD4 T cell count, and y-axis we have HERV-specific CD8 T cell response. There is a direct correlation suggesting that higher the HERV-specific T cell response, higher will be the CD4 positive T cells. So if CD4 cells are going down, if you want to increase, you can say that we should really somehow make this HERV T cell response going up. So it means that HERV-specific T cell response could be doing something great to really having, better CD4 response and lowering the virus down. How can the HIV viral load go down? The only possibility is that if HERV-specific T cell can lyse HIV-infected cells. And when there is no HIV-infected cells, so there won't be any host for HIV to proliferate itself and produce its progeny more and more. That's the basic idea, right? So that's what we have to see what is happening. But conclusion for this part, what you see here is controllers have HERV responses of higher magnitude, breadth than HERV suppressed patients, virological non-controller, immunologic progressors, and HIV negative subject. In untreated patients, there is an inverse correlation between HERV response and HIV-1 viral load. In untreated patients, there is a direct correlation between HERV response and CD4 count, right? Now, as I just said, what is happening, what these HERV-specific T cell responses or T cells are doing. To that, we thought that there is a possibility that we can make HERV T cell based vaccines. What do you say? HERV-K vaccines for HIV. One of the important categories for HERV is called HERV-K. HERV-K. Now, to begin with that, we needed to know whether when you do in vitro infection of HIV, this infection, so far what we did, we just took the patient who got infected, God knows when. But if you take cells, CD4 cells from a person infected with HIV, and in that cells you went to see whether there is a upregulation of HERV or not. You see that different kind of human endogenous retroviruses. CD4 cells was purified by primary HIV. So induction of HERV-K in CD4 cells by primary HIV isolate. We purified CD4 cells and we infected with we infected with HIV isolate, and then we followed, hours, 25 hours, 50 hours, 75 hours. You see the red one. Red one indicates copies of HERV-K GAG per TBP. TBP is a housekeeping gene. It's a real-time PCR study. You see that there is an increase in the copy number of HERV-K and goes down because of cell starts dying because you infected with HIV. Likewise, HERV-K, there is an increase, then decrease. Infection level for HIV, you can see that infection is really happening. Mock, you see nothing is happening. And here you see that upregulation of HERV-K envelope, HERV-K reg, suggesting that even in vitro, if you try to infect with HIV, there is an upregulation of HERV at transcription level. When it is happening, then we wanted to know what T cells might be doing. So the same patient, one of the patient from the previous slide I showed, who had high magnitude of T cell response against HERV, we cultured the T cells from that person. We did a serial dilution and we ended up collecting T cell clone which is against human endogenous retroviruses. And the study was designed, you see that. We took the autologous CD4 T cells. In autologous CD4 T cell, we infected with HIV. From the same person, we took the T cell clone, and we also had as a control HLA mismatched CD4 T cells. We mixed at the ratio of 1:10, effector and target, co-cultured, and then here we also added HIV-1 GAG-specific CD8 T cell clone. This CD8 T cell clone is coming from the same person, so that there is no HLA mismatching here, but here HLA mismatch has been added because of the control, negative control. And then we co-cultured it when the CD8 clone, HIV and HERV, there are three different clones, HERV clone, CD8 HIV GAG clone, and CMVPP65 specific CD8 clone. Three different clones were mixed here, and then if HERV specific T cell is able to lyse the cells, we have to see some readings. What readings? What we see? There will be massive decline in HIV, right? Because when cell lyse, so HIV is gone. HIV is not infecting other cells, so there won't be any expression. With this, when we tried to do flow cytometry, first example is there is no clone. It means that these are what? These are different clones from the same person we prepared. And if there is no clone, so what will happen when HIV infect the cells? You can see HIV expression. And what HIV expression you see here? You see that there is a 48.9% in these CD4 cells HIV GAG expression. You see, that shows that high infection, right? If you add a different clone, it means any CD8 cells which is not specific for HERV, not specific for HIV, this clone will not be able to clear HIV infection at all. So here what you see that the infectivity remains elevated, that is 45.8, where we had a CMVPP65 specific clone. In third situation, when we add HIV specific clone, right? So what will happen? You see that there is a massive decline in HIV positivity. You see the HIV GAG from 49, it has come down to 3.9%. It means that HIV GAG specific clone is able to lyse the infected cells, right? That is what we should expect. But interesting thing is that we need to know what HERV specific clones are doing, whether they are also able to lyse HIV infected cells or not. If they lyse, we should expect very low percentage of HIV infected cells. You see that indeed, when we add HIV GAG specific clone, it also brought down this GAG percentage from 49.9% to 5.85, suggesting that HERV specific clone, CD8 T cell clone, has ability to lyse HIV infected cells. Now the question is that if I make peptide pool or small peptides or a small antigen that represents HERV and inject in a person, the person will start developing T cells against HERV, and they remain in our body for a memory, like a memory T cells. So any HIV comes in the body, infect our cells, it will push HERV to come up, and these cells should attack, and then HIV infection can be prevented. That's actually the hypothesis, that's actually the concept which we had in our mind. And what was that concept? We can explain it using this particular figure. You see that when HIV infects the cells, cell will express two different proteins on its surface. One is HIV, right? Second one is HERV. At this particular time, you develop T cell response against HIV, THIV, and also T-HERV. Both T cell response are there. Both T cell response have ability to lyse HIV infected cells. Initially, actually it happens initially. That is why when you get HIV infection, after 3 weeks after virus enters in the body, you develop primary HIV symptom, fever, lymphadenopathy, rashes and all that. Then slowly you see that you are okay, you are fine, because your immunity has evolved and it is pushing the virus down, and CD4 count is again coming up. You feel good for 7-8 years. Why again you become sick? Because HIV has habit of undergoing mutations. You see that this has changed its sequence. When it has changed the sequence, HIV-specific T cell is no longer able to attack it. But what remains there intact is HERV-specific T cell response, HERV. This is the only T cell response capable of fighting HIV-infected cells, because this HERV is not going to change. So based on this particular concept, we thought that HERV-based T cell vaccine could be rolled out. The work was done. We did it later on people started working on this non-human primates as well, I think in United States, and other retroelements were also explored. But the major challenges, what question mark remains, and that's why people are a little worried about this work, that it may induce possibly autoimmune disorder. Imagine you have this scenario, it is good for HIV, but in the meantime you get some other seasonal allergy or autoimmune disorder and other things where different cells express HERV, so these T cells will start attacking other part as well. So based on that, this is something is still in the process of further evaluation, whether it will be safe to further pursue this vaccine strategy based on the human endogenous retroviruses or not. But this is very exciting thing. What message we get that HERV, what we have in our body, or in general viruses which we are exposed to, somehow are helping us in various different situations. So this is something which is related to HIV. And you see that conclusion for this part is the study demonstrate the example of a specific recognition and elimination of HIV-1 infected cells by anti-HERV T cells, as it is the HERV antigen rather than HIV itself which is recognized by these T cell. Elimination of infected cells occur irrespective of the sequence variation in HIV, right? Vaccine-induced HERV T cell response may play an important role in containment of HIV-1, right? This is something about human endogenous, but as I said that there are 10 to power 31 viruses. What about the other viruses? Anything would like to ask at this point, because now I'm changing the story bit. We are just talking about human virome rather than human endogenous retrovirus alone. Yes, please.
Just 2 slides previous where you explained the HIV-K expression with the housekeeping gene. In the third graph, we can arise some point of housekeeping gene. Can you elaborate this area? Yes, second graph.
Which one?
K positive.
What?
Housekeeping gene से थोड़ा सा नीचे blue light.
No, it is not. This is a mock. This has nothing to do with housekeeping. Housekeeping gene is used to just, control. This is a qPCR based study. Housekeeping genes are those genes where expression is constitutively expressed. Suppose you add various different concentration of the RNA template. So results may vary. But if you just take, let's say, one calibrator, let's say you take a housekeeping gene. So when you compare it with per copy of housekeeping gene, then result get normalized. Housekeeping gene are used for the normalization. The blue line that you see is a mock uninfected. This mock suggests that there is no upregulation of human endogenous retrovirus. Only HIV is doing that. The further decrease in this HIV HERV expression is because of cells start dying because you infected with HIV, right? This is explanation for this, right?
Sir?
Anyone else? हाँ बताइए.
HRV transcript T cells, that is CD5 receptor, that is also dysregulated in T organisms. So it's like T cells are getting dysregulated and causing cancer. It can also be used as a treatment
Which one? Sorry, just be louder a little bit. You are asking about certain different other marker.
CD5 जो T cell का marker है, वो HCl की E-derived marker है, जो T all से regulated है, मतलब T cell acute leukemia. हम उसको भी तो as a therapy use कर सकते हैं.
We can do. You see that. It's good you raised this point because I just specifically talked about HERV in the setting of HIV. You see that syncytia formation during the placenta development in the fetus is driven by HERV-W. HERV-W is very, very essential. If you completely knock down with this HERV, so there will be a problem to conceive as well. HERV has several benefits. It has been shown that there are certain proteins derived from HERVs which are responsible for us to retain memories. You know that you had gone to some other place. You will be knowing this. You had this lecture after even 5 years, something about how do you remember all these things. So it has been documented, and there was a paper, I think, in Nature of Science, which talks about HERV-derived proteins are there, which transmit from cell to cells and retain as a memory. So many things are there. But you are asking that has to be documented whether it is really having the same kind of thing, because what here I am showing the T cells is specifically for HERV-K envelope is able to lyse it. If I isolate T cells against the same HERV you are talking about, showing exactly the same thing, why not? It can also be used. Since it's a humongous amount of work, we ended up just taking HERV-K because we found HIV-infected person who had the highest magnitude of T cell response against HERV. So I thought that when the person is having the highest response, so why not choose this one to just, to make a proof of concept. This study needs to be further investigated, taking a different HERVs and then T cell clone and see whether the lysis is happening exactly the same efficient way, the way it is happening in case of HERV-K envelope. So why not? Yes. Okay, so shall we go ahead? So now, virome, we talk about bacterium, and gut microbiome, we talk about the various different kind of bacteria, firmicutes, bacteroidetes and all that. Right? But what about virome? See that if you look at the entire different kind of anatomical region in our body, you see that the human virome consists of a various species of viruses which are present in our body. What are the different places where it can have? We see the nervous system, mouth, the maternal male respiratory tract, urinary tract, blood, joint, GI tract, everywhere you have various different kind of viruses. They're not causing anything, right? A step-by-step assembly of neonatal virome. You see that month 0, and then you see that kid playing on the floor, taking everything on mouth. What do you think? A lot of viruses are going inside our body. And they develop different sets of viruses. Phages are more there because all the bacteria, everything that child is exposed to avoid the colonization of pathogenic bacteria. So nature has evolved our body in such a way that we got a lot of bacteriophages as well, so that they stop the growth of pathogenic bacteria, right? Viruses of human cells, all these things. You see that factors that shape the human virome, it's breastfeeding, diet, genetic, aging, disease, geography, cohabitation, medication, so many things are there. So the take-home message is that we have certain different kind of viral set in our body that include bacteriophage, endogenous retrovirus, for which I talked about, non-pathogenic RNA virus, pathogenic RNA virus, pathogenic DNA virus, non-pathogenic DNA viruses. So we are full of viruses. You see that? This is a very interesting one. This slide shows different kind of viral families. You see that the brain, herpes virus, virus, flavivirus in blood, all different kind of DNA viruses, RNA viruses. I'm not reading, but you'll be amazed to know that name any virus, a lot of viral transcript you have in your body. Full of human body is a home to diverse viral species under non-pathological condition. That's important thing. Whenever there is a pathological conditions, whenever you have viral infection, you just get rid of it in 7 days bed rest and all that. I'm not talking about serious viral infection which happened recently, COVID-19 and all that, but most of the viral infection has the same kind of thing. You see that intestinal bacteriophages. So I said that our intestine, lot of bacteria is there, but there are a lot of bacteriophages there. You see 10x more viral particle than bacteria in feces, 10 times. If you look at the bacteria in fecal material, and you should expect there are 10 times more bacteriophages in the stool than the bacteria. High level of genetic diversity, source of transfer of genes between bacteria. You see, this is also very interesting. 2 different bacteria are there, they can transfer the genome. We studied about this transduction and all that, during our BSc. So bactericidal due to lytic replication. You see here, you have invertebrates and vertebrates, and you have environment and mucus, and there is a comparison, phage to bacteria ratio. You see that phage to bacteria ratio is always higher in mucus than environment. So environment this ratio is not that great if you compare the human being, the mucous membrane. So whether it is human gum, you see that here, or mouse intestine here. So this is very interesting thing. But when you look at the prevalence of human DNA viruses, the study was conducted in 2017. The prevalence of human DNA viruses in 8,240 individuals. You see that so many different viruses were reported. This is the prevalence. You see that? It's interesting to know. One important thing is that immunologically, I would like to clear, if you see any viral transcript using PCR or next generation sequencing, it doesn't mean always that there is a live virus present there. It could only be the transcript which is floating inside the blood. Because when the virus is there, virus dies, what happens? The nucleic acid remains there, which you can detect. The true assay for the presence of virus is the virus isolation, where you look for its infectivity. Anyway, that's a different topic, but I just wanted to let you know, because the same kind of that discussion came in the picture when the people started reporting that in sewage also there was a COVID-19 transcript when they did a qPCR, right? But the presence of that COVID-19 transcript in the sewage water did not really confirm whether you could isolate virus from there, because when you try using it to infect cells, you may not be able to infect because only transcript was there. So virus isolation is something very important. So as I just mentioned previously, HIV in the era of antiretroviral therapy, what happens when a person is on antiretroviral therapy? There is a undetectable viral load for sure, preserved CD4 count, but still they have exhausted T cells, they have senescent T cells. T cell exhaustion and T cell senescence are responsible for causing certain organ dysfunction and in extreme cases organ damage because of the high inflammation. Because despite suppressive therapy, HIV infected urinary tract, blood, joint, GI tract, everywhere you have various different kind of viruses. They're not causing anything, right? A stepwise assembly of neonatal virome. You see that month 0, and then you see that kid playing on the floor, taking everything on mouth. What do you think? A lot of viruses are going inside our body. And they develop different sets of viruses. Phages are more there because all the bacteria, everything that child is exposed to avoid the colonization of pathogenic bacteria. So nature has evolved our body in such a way that we got a lot of bacteriophages as well, so that they stop the growth of pathogenic bacteria, right? Viruses of human cells, all these things. You see that factor that shape the human virome. It's a breastfeeding, diet, genetic, aging, disease, geography, cohabitation, medication. So many things are there. So the take-home message is that we have certain different kind of viral set in our body that include bacteriophage, endogenous retrovirus, for which I talked about, non-pathogenic RNA virus, pathogenic RNA virus, pathogenic DNA virus, non-pathogenic DNA viruses. So we are full of viruses. You see that? This is a very interesting one. This slide shows different kind of viral families. You see that the brain, herpes virus, virus, flavivirus in blood, all different kind of DNA viruses, RNA viruses. I'm not reading, but you'll be amazed to know that name any virus, a lot of viral transcript you have in your body. Full of human body is a home to diverse viral species under non-pathological condition. That's an important thing. Whenever there is a pathological conditions, whenever you have viral infection, you just get rid of it in 7 days bed rest and all that. I'm not talking about serious viral infection which happened recently, COVID-19 and all that, but most of the viral infection has the same kind of thing. You see that intestinal bacteriophages. So I said that our intestine, lot of bacteria is there, but there are a lot of bacteriophages there. You see 10x more viral particle than bacteria in feces, 10 times. If you look at the bacteria in fecal material, and you should expect there are 10 times more bacteriophages in the stool than the bacteria. High level of genetic diversity, source of transfer of genes between bacteria. You see, this is also very interesting. 2 different bacteria are there, they can transfer the genome. We studied about this transduction and all that, during our BSc. So bactericidal due to lytic replication. You see here, you have invertebrates and vertebrates, and you have environment and mucus, and there is a comparison. Phage to bacteria ratio. You see that? Phage to bacteria ratio is always higher in mucus than environment. So environment this ratio is not that great if you compare the human being, the mucous membrane. So whether it is human gum, you see that here, or mouse intestine here. So this is very interesting thing. But when you look at the prevalence of human DNA viruses, the study was conducted in 2017. The prevalence of human DNA viruses in 8,240 individuals. You see that so many different viruses were reported. This is the prevalence. You see that? It's interesting to know. One important thing is that immunologically, I would like to clear, if you see any viral transcript using PCR or next generation sequencing, it doesn't mean always that there is a live virus present there. It could only be the transcript which is floating inside the blood. Because when the virus is there, virus dies, what happens? The nucleic acid remains there, which you can detect. The true assay for the presence of virus is the virus isolation, where you look for its infectivity. Anyway, that's a different topic, but I just wanted to let you know, because the same kind of that discussion came in the picture when the people started reporting that in sewage also there was a COVID-19 transcript when they did qPCR, right? But the presence of that COVID-19 transcript in the sewage water did not really confirm whether you can isolate virus from there, because when you try using it to infect cells, you may not be able to infect because only transcript was there. So virus isolation is something very important. So as I just mentioned previously, HIV in the era of antiretroviral therapy, what happens when a person is on antiretroviral therapy? There is a undetectable viral load for sure, preserved CD4 count, but still they have exhausted T cells, they have senescent T cells. T cell exhaustion and T cell senescence are responsible for causing certain organ dysfunction and in extreme cases organ damage because of the high inflammation. Because despite suppressive therapy, HIV infected subjects showed that certain bacterial population which we call gut dysbiosis, happens during HIV infection that sustain even during the treatment. And this dysbiosis is associated with the inflammation. If you think about the gut of an individual, where most of the bacteria and most of the TCD4 cells there, because in gut there are gut-associated lymphoid tissue. In gut-associated lymphoid tissue, this is something secondary lymphoid tissue like lymph node. Lot of CD4 cells are there. HIV infection happens, CD4 cells goes down, gut homeostasis changes, gut microbiome changes. The moment gut microbiome changes, so inside the gut there are gut epithelial cells which are attached to each other through tight junctions protein. When this dysbiosis happens, there is a disruption in the tight junctions protein there. When there is a disruption in the tight junctions protein, then you have a leaky gut. Through this gut, microbial product can translocate from intestine, gut to the peripheral blood. When they come to the peripheral blood, they interact with the macrophages. They interact with other immune cells. Through TLR and all that, they start secreting pro-inflammatory cytokine. These pro-inflammatory cytokine, now they remains elevated in our blood, peripheral blood, and long time exposure of these pro-inflammatory cytokine causes issues with our vital organ. It leads to non-AIDS morbidities. Heart disease, kidney disease, neurocognitive impairment, all these kind of things are happening. So people found that there is a dysbiosis. People thought, let's try to give some probiotics so that the gut microbiome could, we could reverse the gut microbiome. For dysbiosis, we should expect eubiosis, where a lot of good bacteria are there. That work is still, people are working on that, and in certain cases it is promising as well. But in addition to this, no one actually thought of looking at the bacterial population that is changing. When dysbiosis is happening, what is happening to the viral component? We do not know. Another thing is that when we talk about non-AIDS morbidity, so inflammation is happening in the peripheral blood. Why? Microbial component is coming. This microbial component is interacting with your immune cells. But if there are some viral component present in your blood and viral nucleic acid is there, that nucleic acid could also be sensed by various different kind of immune cells, leading to the inflammation. So we conducted a pilot study. In this pilot study, small HIV group of HIV infected individual, we took patients who are on heart. Why did we select people who are on antiretroviral therapy? Because nowadays, whenever a person is detected as HIV positive, immediately the person is put on medication. So whatever people you get in ART center, in AIM, Safdarjung or any hospital, they are all on therapy. So our target population as of now present is HIV infected people who are on antiretroviral therapy, that is ART. So we have to see what problem they are actually facing. So our target is ART. So we took HIV patient on ART. We took healthy control. What we did, we took blood sample. In blood sample, we purified plasma. We also had PBMCs. In the plasma, we started looking at the virome composition. Very different kind of viruses are there. We were surprised. I would expect HIV is there, but in addition to HIV, majority of non-pathogenic bacteria, non-pathogenic viruses that we have, they are DNA viruses. So we aim particularly non-pathogenic viruses, DNA viruses, and we found that in both in HIV as well as control group, various different families of virus could be obtained through next generation sequencing. Viral metagenomics approach, we found several different families of the viruses present not only in healthy control, also in HIV infected subject. Now, we wanted to know whether there is a differences in the relative abundance of these families or species of the viruses. comparative viral diversity, richness, and evenness. So, one of the important criteria to find out the diversity is looking at the Shannon diversity and Chow-1 index. So you see that HIV-1 infected subject here also, the richness, Chow-1 and Simpson. What we actually found, there is a significant differences in the diversity of the viral family present in HIV infected person on therapy compared to controls. You see, we ended up publishing it 2024 as well. And then, since it was a pilot study, small sample size, we are just expanding it with more people, more different category, and we found relative abundance of viral family in any species. So gamma herpes viruses, human gamma herpes viruses 4, we found significantly elevated level of or relative abundance of herpes gamma retroviruses 4 in HIV infected subject. Herpes viridae also, there was a relatively significant high abundance, and also we have C4 viridae. So significantly elevated relative abundance of C4 viridae in HIV infected subjects on ART compared to that of healthy controls. You see that some bioinformatics analysis with this cladogram, we showed that there are certain different taxa of the virus which are significantly abundant in ART suppressed HIV infected adults. So these are very important. When C4 viridae, human gamma herpes virus 4, and lympho cryptoviruses, herpes viridales, herpes viricities, papilloviricota, and herpes viridae. So these are important ones. And now, the important thing is that we needed to know what these different kind of viral taxa are doing in HIV infected persons. Whether they are associated with the T cell dysfunction. In T cell dysfunction, where we talked about T cell senescence or T cell exhaustion. Third category is inflammation, by looking at inflammatory cytokine in the blood. So you see, we further looked at T cell senescence. When you age, your T cells gets not basically exhausted, we call T cell senescence. When you age, your T cell also age with your age. One of the important marker for senescent T cells among elderly is the presence of CD57 and absence of CD28. So you see that when I take PBMC of elderly person and I stain these T cells for the presence of CD57 and CD28, so higher magnitude or higher percentage of CD57 positive and CD28 negative represents higher level of T cell senescent. Higher level of T cell senescent is associated with high inflammation in elderly. HIV infection results in the establishment of T cell senescent at very early stage. Imagine like a 35-year-old HIV infected person may represent the same senescent level of the T cell as that of like a 60, 65 or 70-year-old person. So in this way, people also say that accelerated aging is a hallmark of HIV infection. So all different kind of non-AIDS complication people experience during HIV infection are exactly the same complication experienced by elderly. So that is why people say that accelerated aging is a hallmark of HIV infections, right? It is just because of inflammation. The same kind of concept which I explained for the leaky gut is also happening in case of elderly. Those people, like, normally without any HIV infection, when they age, aging also results in the changes in the gut microbiome. Whenever there is a dysbiosis, it will cause impairment of the tight junctions protein existing between gut epithelial cells. That makes these things like a leaky gut. When you have a leaky gut, there are chances that the microbial product translocate from your gut into the peripheral blood, and that induce inflammation happening in the case of elderly as well. That's what people say, that eat a lot of yogurt and probiotics and all that, so their gut is very good. And if the gut is good, then you have a healthy aging. Gut is not good, then you have unhealthy aging, because it gives rise to various different kind of inflammatory diseases. Diabetes is one of them. And people, you may have experienced that, and sometimes people have a lot of sugary thing, but still they are thin. And many people, they just do a lot of exercise, they avoid, certain fatty food and all that, but still they complain that, I've done so many things, but my weight is not getting reduced. So just think about their microbiome, which is causing them. So this has been, published also, and people know about it. So this, anyway. So here you see, this is a gating strategy. I'm not getting into it, but what I'm showing here, the CD57 positive CD8 T cells, you see it's significantly elevated in case of HIV infected people who are on antiretroviral therapy compared to the HIV negative control. And this is CD8 T cells. And then we also looked at various different kind of markers which are associated with T cell exhaustion. You keep running, you start sweating, and then you are exhausted. You no longer efficiently run. The same thing is highly present on the surface of CD8 T cells. You mark these T cells as exhausted T cells. So this is a gating strategy for immune exhaustion in the treated HIV patient in cohort. And then we know the frequency of T cells, which is senescent T cells, which are exhausted T cells. We wanted to know whether in any way they are associated with a different kind of viral taxa or not. Then using this R program, using R software, you see that, we ended up actually correlating various different kind of viral taxa with various different kind of T cells phenotypes. So a plasma virome correlation with immunosenescence and immune exhaustion in HIV infected individual on ART, we found that human gamma herpes virus 4, which was elevated, we found in HIV infected patient, is having association with PD1 positive CD4 cells. Likewise, you see that we have CTLA4 minus and TIM3 positive CD8 T cells. Various different kind of T cells which are considered as exhausted T cell are associated with herpes, human gamma herpes virus 4, which was elevated in HIV infected subjects. Correlation pattern virus with the T cell immunophenotyping. Here again you see that exhausted T cells, various different kind of virus. This continues because the data is so big. So I'm just not really going one virus species at a time, just trying to give you the overview how these virome composition is associated with different phenotype of the T cells. And other thing was that the plasma pro-inflammatory cytokines, you see that IL-8 is elevated in HIV infected people who are on antiretroviral therapy. TNF-alpha is again elevated here. And when we try to correlate it with various different kind of viruses with plasma cytokines, pro-inflammatory cytokine, you see that human gamma herpes virus 4, again, it is positively correlated with human TNF-alpha. Likewise, Staphylococcus fages as well, and herpes viridae, these are also positively associated, suggesting that any viral composition, if it changes in HIV infection, it is associated with the pro-inflammatory signature in the blood. It is very important that we should extensively study the complete set of the virome. This work was done on the plasma, and plasma should not be considered as a site where you could locate different viruses or bacteria, but indeed people are reporting these days. Not saying that is a live virus or something, even their transcript can cause different alteration in the immune responses. The next stage, what we are planning to do is looking at the gut virome composition, because whatever you see in the plasma is coming from the gut itself. So when we have a full picture of the gut virome, then we would be able to say completely what are actually causing these viral translocation, like a bacterial translocation and the viral translocation, and then we compare the relative abundance of a particular viral taxa in the gut with that of plasma. And we do more study because this was a pilot study, and we are just conducting the big study where we can include more patient and more study will be conducted. So what conclusion we get out of it is human virome is unexplored area of research. As I said in the beginning, virome composition and HERV modulate the immune system. Viral species, human, gamma, herpes, viruses 4, and viral family herpes viridae correlated positively with cytokine TNF-alpha in HIV-infected individual on ART. Cytokines IL-8 and TNF-alpha significantly elevated in HIV-infected individual on ART. Plasma virome, as I said, that we do not know whether there is a live virus. We believe there is a viral transcript floating in peripheral blood. If it is happening, so plasma virome can activate immune system through different kind of pattern recognition receptors. What are pattern recognition receptors? You may be knowing various different TLRs. When they get inside the cells, they can also interact with RIG-like receptor. So when they interact with pattern recognition receptor, they elicit pro-inflammatory responses, and these responses may actually make a person with high inflammation depending on what kind of virus is there in your plasma, viral transcript you have in the plasma, and what is its load compared to the healthy individual. So comprehensive plasma, salivary and gut virome study with a larger cohort is needed for better understanding so far. So these are the things which I wanted to discuss. And now, any doubt with the people back in San Francisco, UCSF and University of Toronto and Jacobi Medical Center, Michael G. Rosenberg, and later on was plasma virome study, which I showed here. It's part of the lab where I'm working now in Delhi. So that's like a 2 different set of the data I showed you. So I'd like to acknowledge Tanu Bagchandani, my previous PhD student, Dr. Ankita Rai. Anjali Verma recently submitted her thesis. Nikita will be submitting her thesis next year. Himanshu is a nodal officer ART Center in King George Medical University in Lucknow. Douglas Nixon was my mentor, postdoc mentor, who was really inspired me to work on human endogenous retroviruses together with Mario Ostrowski in University of Toronto. And children cohort came from Michael G. Rosenberg. And of course, patients are very important, so I should also acknowledge them. And of course, ANRF motivated me to continue work on this particular area. I acknowledge JNU Pair Program, ICMR, UGC. So now I'm very happy to take any question very quickly because there is a Viva going to happen very soon. Thank you for listening. Anything, any thought you have, कुछ आपको नई चीज़ पता लगी हो आज के उससे, just share few things. हाँ बताएं.
Like you talked about the T-cell senescence and inversion and the expression of CTLA4 and PD-1. Is there any relation of T regulatory cells as if these expression will lead to the cancer? Like these are the...
Well, I didn't talk about cancer, but I can tell you one important thing in general about T regulatory cells. In blood, immunological parameter which decides how the progress of disease will take place is a Th1 by Th2 ratio. Likewise, in guts, Th17 by T regulatory ratio is very, very important. Th17 is very, very important. As we see during aging or in HIV infection, there is a decline in Th17. When there is decline in Th17, the share of T regulatory cell by default increases. So higher T regulatory cells during HIV infection sometimes supports HIV progression, because it stops those important HIV-specific T cells to act against HIV-infected cells, because T regulatory cells function is what? It stops the proliferation of antigen-specific T cell response. So this is, I can say that. I didn't actually specifically look at relation existing between herb, any herb or any different kind of viral taxa with that of T regulatory cells, but it can be studied. But in general, in the setting of HIV, what ratio is important, I just mentioned. It's a Th17 by T reg ratios. That's important. Anyone else?